Hello, welcome back. I'm missing a nail, but otherwise we are intact, and today we are talking about cell membranes, membrane fluidity, membrane components. Very cool. And then next time we're going to be talking about what is a cell, what's in a cell, and then you'll see why membrane fluidity is so important and all the little structures. Okay, so we can just say cell membranes, because that's what we're flicking talking about. And you do not have to make it fancy like me, but I do enjoy it. This is why studying is fun, guys, for aesthetics. Anyway, so I'm sure you remember all of the things that we talked about last time with our lovely macromolecules. We had some lipids, some carbohydrates, etc. But if you remember, when we talked earlier, we talked about some lovely things called phospholipids. Let's see if we can find them. Phospholipids, remember this general structure, and also saturated versus unsaturated fatty acids. So just as a reminder, we have a phospholipid. It has a phosphate group, PO4 negative. And then these are like the hydrocarbon chains. Remember how we had like the chemical structure where like each like bend in the line was a carbon molecule or a carbon atom. So you have like each bend representing a carbon with hydrogens attached to fill the octet. You don't remember, this is not OChem. You don't really have to do this. You don't even have to draw it. But it is nice to know. So these are hydrocarbon chains. And so this is your lovely phospholipid. So this is your polar head and your nonpolar tail. And this is polar, like it's charged now. So it's going to really be attracted to things like water, which is polar, and the nonpolar tail is not. So when you have your cell membrane, which we're going to draw in a second, but we're not going to draw it yet, all the nonpolar tails are going to face each other. You're going to have, in cell membranes, it's a layer of two, we can draw this. In your, this phosphate group is smaller. PO4. There we go. Nope, pretend that's nice and even. But the nonpolar tails are going to face inward, because out here is like, say this is inside the cell and this is outside the cell. Think your cell is huge, right? And so this is the entire inside of the cell, and this is like out, the extracellular matrix. Both sides are going to have water. Like the inside the cell is going to have water, outside the cell is going to have water. So you're going to have like a water molecule. Like if I were to draw it like this, H2O. We have the partially negative oxygen, because, it's more electronegative. It's going to be taking, like, the electron density away from the hydrogens. So it's going to be partially negative, and the hydrogens are going to be partially positive. That's what these little symbols mean. It's like partially. So, like a partial charge. So this hydrogen is going to be partially positive, too. So it's going to be attracted to this negative phosphate group. The same thing goes over here. We're going to have another water molecule where the hydrogens are kind of attracted to the negatively charged phosphate group. So that's just kind of like the general structure of the cell membrane. That's how you're always going to see it being drawn. And remember, these fatty acid tails, the way I drew them, they're a saturated fatty acid. There's no double bonds. But if it were double bonded, then it would be, it could make it a cis saturated fatty acid, and then it would point like downward, and then you could get like less cramming, which is what we're going to talk about now. So in your cell membrane, you're going to have, it's going to be made up of these it's called a phospholipid bilayer. So phospholipid, one of these guys, we can say phospholipid, that's who this is. And since there's two of them, which make up a layer, it's a phospholipid bilayer. Wow. So there's two components of membrane fluidity. So, how fluid your membrane is, cell membrane. And that's going to be, one, how tightly packed your components are. And two, how much movement there is. So when people talk about like cell membranes in biology, they they say this thing called a fluid mosaic model, which is basically the idea that like here, I'll just show like this is basically like a very simplified diagram. So imagine this is your phospholipid head, and this is the the phosphate group, and then these are the like fatty acid chains. This is a very simplified diagram of like one like cross section of your phospholipid bilayer. This is how it's drawn a lot in like simplified diagrams. So here, your extracellular matrix, polar, full of water, attracted to the polar charged heads, and then intracellular matrix, also full of water, attracted to the polar charged phosphate groups, same thing, and then that pushes the nonpolar stuff inside. We kind of talked about that like idea where the nonpolar stuff gets pushed together when we talked about proteins earlier. So basically this is our general structure, and the fluid mosaic model is like all of these lipids are kind of like sloshing around like the ocean, and there's like all this stuff also in the membranes that kind of just like float around in there. That's just like a term that you'll hear, but that's what it's referring to. So the membrane, it's going to have all of these phospholipids, and say your membrane isn't fluid enough, it's like really rigid. Well, you could replace the phospholipids with unsaturated fats. So, unsaturated fats replace phospholipids. Because remember, they have those double bonds that can take up space, and that'll make it more fluid because it's less crammed. Which take up more space. Well, I wouldn't say which. I'll say and take up more space, because the phospholipids take up less space, so you can cram more of them in there, but then it's like crammed, So I'll say and. That was not a very good whiteout job. That's okay. And take up more space. Which increases fluidity. So this will happen when your cell membrane is too rigid. And this can happen like with a variety of circumstances. But for example, when your cell is experiencing higher temperatures, the fluidity will increase. So if the temperature is high and then it's chilling, but then it lowers and then it becomes less fluid, maybe we'll replace some of the phospholipids with unsaturated fats to like regulate, keep that general level of fluidity. So like this will happen when the membrane is too rigid. And let's make a note for ourselves. Higher temperature equals higher fluidity. Okay, cool. I'm gonna make a little thought bubble here, just like a don't forget, But also, higher unsaturated fats will be higher fluidity. So maybe your temperature decreases, you'll increase the number of unsaturated fats. Makes sense. Okay, but you can also, instead of unsaturated fats, maybe you want like a temporary fix. This is definitely if you're changing temperature. Add in cholesterol. Do we remember what cholesterol looks like? It was, like 3 rings in a doghouse? General structure. We'll draw it in our little diagram later, but I can draw a little. It's like 6 carbon rings. You don't need to know this. What is it, like this? Something like that. Oh, I think. Yeah, cholesterol, generally, I think. So cholesterol, you can add that into the membrane, which takes up space. Same thing. And it can kind of depend. The cholesterol is going to increase fluidity usually less than an unsaturated fat. But more importantly, it's kind of a temporary fix. So this takes up space, similar kind of idea, and increase fluidity. Okay, so just to summarize, say we want to increase fluidity. So just to recap, there's a couple ways we can increase fluidity. Okay, so unsaturated fats, we know that's going to increase fluidity. So if you increase the amount of unsaturated fats in the phospholipid bilayer, you're going to increase the fluidity of the membrane. But also if you increase the temperature, that's going to increase fluidity. And finally, cholesterol. Cholesterol. Cool. So these are generally like good ways to increase your fluidity. So our cholesterol is kind of like a buffer because it'll increase fluidity with phospholipids, more fluid than a phospholipid, but less fluid than an unsaturated fat. You can make a note of that if you want. Cholesterol is a buffer. So if you're going to say like on the fluidity scale, so like most fluid to like less fluid, we can say most fluidity. Okay, less fluidity. Kind of consistent here, but that's okay. We'll have our phospholipids here. Phospholipids. They're going to be the least fluid. That's like our typical, we can really cram them in there. And then our unsaturated fats. They're pretty fluid. And then, so our cholesterol is going to be right there in the middle. Maybe not directly in the middle, but it's in between. That's the point. So those are our points. You can increase cholesterol, increase fluidity when you got lots of phospholipids, or the other way if you have lots of unsaturated fats. And then we can color these. Same color as our phosphate groups that are polar. They're charged, not just polar, but charged. Okay, little review. What affects membrane fluidity, how tightly packed the components are. That's why phospholipids are like less fluid because they can get real packed because their little straight hydrocarbon tails can get really crammed in there, unlike an unsaturated fat, which is like go all over the place. And then how much movement there is. Okay, so unsaturated fats can replace phospholipids and take up more space. That's really important. I guess replace phospholipids is important too. And that increases fluidity. And this is you'll do this when the membrane is too rigid. And so addition of cholesterol will take up more space as well, and that can also increase fluidity, especially in comparison to your phospholipids. So we can take a little note. This is where our like relative fluidity is. Okay, cool. And then we can just say, these are the things that increase fluidity. Cool. Okay, so we're still doing cell membranes, so we can write another header. But this time we're going to draw like a big diagram of this thing with all the stuff we might have in it. Because turns out cell membranes aren't just phospholipids. That's like the canvas, that's like the background. But there's other stuff in there too. So obviously when we have a cell, it's going to be like interacting with other cells and like having an environment and stuff. And so you want to keep a cell contained, especially animal cells. They don't have a cell wall. So the cell membranes are what keeps it together. So they're very important. They got a lot of stuff going on. So we can just draw a bunch of phospholipids. Actually, I'm going to draw them like this. So let's say we have a bunch of phospholipids, and these are the little tails. I'm just simplifying them like I did over here. And then we're going to have like a thing sticking out, and we'll talk about what this thing is later. But right now it's just like a shoot, basically. Like imagine it's being cut, We're taking a cross-section of a cell. So we got all these phospholipids. I guess the tails are like kind of long because it's like kind of touching. Okay, maybe these aren't long enough. Imagine they're longer, like this. And they're like best friends. And that's how you get your cell membrane. Okay, so this is a protein in the cell wall. Bet you didn't know that could happen, but it can. but we still have more phospholipids going on. And then maybe we have cholesterol. We'll draw her correctly, except actually not really. Oh, this should be, yeah, that's like that. Cholesterol's structure is a little bit more complex than we need to know, but usually it'll be like this. It'll be, so 3 6-carbon rings, and then 1 5-carbon ring, so like that. So I don't know the exact scale, but this one's kind of taking up a lot of space, making it maybe a little bit more fluid. And then we have like a big guy here. We'll label this in a second. And then maybe a couple more phospholipids. Oh wait, they're supposed to be long, I forgot. They're long guys, okay. And then we'll have more phospholipids over here, because it's a bilayer, meaning 2. We can have another guy. Okay, who are these guys? Actually, no, one more guy. We'll have a guy on the outside. Okay, so this, on the outside, we're going to say this is a peripheral protein. Peripheral protein. So this will be on the inside of the cell, and this will be on the outside of the cell. So inside, outside. Okay. And so say you're like so this is just, you don't really need to know what this guy's doing, but this is a peripheral protein. He's just like on the periphery of the membrane, right? But these guys are in the membrane, like fully embedded within. So it's an integral protein. It's integrated in the membrane. This guy too. We can even color code them. Well, not really color code them, but just like distinguish them from all their glycolipid friends. Glycolipid, phospholipid. What am I talking about? Well, I'm talking about, what I'm thinking about is a glycolipid. So what's a glycolipid? Okay, let's say we have a bunch of carbons. So these are cycloalkanes, just some little OChem stuff for you, but you don't really need to know about that. So say we have like this long chain of carbons, and say this guy has like another guy over here. Let's put him over here. And you're like, what is the point of this? Okay, so this whole situation, like this whole thing, this whole guy, you know, it includes this guy. We can outline him too, so you're like, who? This guy. So this is a glycolipid. So what is the point of this guy? Okay, so this is a hydrocarbon. This is a polysaccharide, actually, because remember how we're talking about all of our starches, you know, our monosaccharide, polysaccharide. So if we have one saccharide unit, maybe it would look like this. Well, we have many, so it's a polysaccharide, and it's attached to a phospholipid. So we have phospholipid plus polysaccharide. That's who this guy is. And what does it do? It's similar to a glycoprotein, and that also has, so we'll talk about a glycoprotein in a second, but there's just so much that they have in common that maybe I should talk about this guy too. So it's very similar. A glycoprotein is basically the same as a glycolipid, except it's a glycoprotein, so it's attached to a protein instead of a lipid. Wow, that's so insightful. Okay, so kind of similar thing going on here. You have your phospholipid in the membrane. You have a protein in the membrane. So this will be your glycoprotein. My page is not very large. Glycoprotein. Okay, so what is that going to do? So both of these have these polysaccharides, and the amount of like carbon bits aren't particularly precise because they're going to vary probably from one glycoprotein to another, because they're like cell ID tags. Remember, this is the outside of the cell, right? This is the outside, this is the inside. So you're going to have other cells. There's a cell over here, right? And they're going to say, who are you? Are you one of our cells? Are you like a bacterial cell? or who are you? Why are you here? And you're going to be like, oh, my glycoprotein is like a cell ID tag, so it can read this. Oh, I don't know why I wrote this in all caps. Cell ID. It'll say cell ID tag, I'll say. And it'll say, hey, glycoprotein, and the glycoprotein will be like, hey guys, I am you. Don't kill me. I'm one of you guys. Because we have so many cells in our body and we have like infections all the time. So how is a cell to know if it's me or you or someone else? So this kind of gives a little bit of insight, you know? Who am I? So it's an ID tag. And so the same thing happens with a glycolipid. So same thing, it's also a cell ID tag. But it also, glycoproteins have a particular, they play an extra role, like particularly in tissue formation. So I'm just going to say, play a role in tissue formation. Hopefully you can read that. Basically what's happening is like, when you're making a tissue, you're going to have a bunch of cells of a certain type, and so you can read this tag and be like, oh, hey guys, I'm just like you. We're going to make a tissue. Super cool. And this is also a protein, believe it or not. And you could say, okay, that's going across the membrane. Maybe you could call it a transmembrane protein. Really insightful name, I know. Okay, so these transmembrane proteins, basically what they're doing is we'll take a molecule from outside or inside the cell, and it'll put it one way or another. So there's a lot of ways it'll do this. But basically what you have to do, and we'll get into it, they transport molecules. Well, they do transport stuff, really. But we'll say molecules. It can sometimes be ions. Okay, we'll say molecules/ions. You can write stuff. That's fine. Both actively and passively. And you're like, what does that mean? We're going to talk about it. Okay, so there's 3 types of transmembrane protein. So this one, I don't know what this one is, but it could be a carrier, which is one way. So a carrier will send like an ion or something one way. So maybe it only pumps from outside to inside. And it also only does, it has binding sites. So it's one way with binding sites. So maybe it'll like accept something here and then be like, okay, now I'm ready. I'm going to shoot it out down here after it bound with the actual protein. Or maybe it'll be something like a channel, which are both ways. What a channel will do for sure is it can let a certain molecule go one way or another depending on the concentration gradient. And you're like, what is that? Well, we're going to talk about it. But also we have receptors. When we talk about signal transduction, these are a huge, huge part of cell communication. So maybe we'll have a protein. When we talked about the importance of proteins earlier in our last lecture, I drew like a little guy where it was like sticking out, and you can have like a little message basically. So we'll have like a little molecule that's traveling through, and then the protein receptor will like attach to the molecule, and then once it gets that message, it'll be like, oh, hey guys, did you know something? I don't know, some sort of message. And the cellular messengers, guys, are called ligands. So it'll accept signals, aka ligands, and sends signals to the cell. So once it accepts this ligand, it'll transmit that message. And we're going to talk about that a lot more soon. Okay, real quick, we're going to talk about entropy. If you haven't heard about that before, entropy is the disorder of the universe. And you're like, what does that have to do with biology? It's okay. All will make sense shortly. So with entropy, we have found, just like through observation and whatever science and stuff, the entropy of anything always likes to increase. So say you have like a tub of water and then you drop a single drop of food dye in it. Do you think it's just going to like sit there and like stay in the exact drop that you put it in? No, it's going to spread out and dye the whole tub of water, whatever color you dyed it. Because it's very concentrated in that drop, right? And it wants to spread out. And that's just generally how the universe works, right? So there's just this thing that we gave a name for it. It's entropy. And for that reason, if you have like a bunch of water on the outside of a cell, and like very little water on the inside, it's going to want to flow in. So we can see that happening, and that's really important for use of energy. So water will want to flow into the cell if there's less water inside. So that doesn't require any energy. It'll actually release energy if it'll like increase the entropy of the universe, right? Or of the cell, whatever. So if we say we don't want any water in the cell, cells don't typically want zero water, but if they did, it would require energy to push it out. So we're going to talk about two types of transport. One using energy that pushes it out against entropy, which requires energy. So if you were to like take the dye and instead of spreading it out, you'd try to like extract it, that would be difficult. That would require energy. Versus just letting it flow, that would release energy. That would be pretty easy. So let's say we just let the water flow into the cell if there's not a lot of water. This won't always be water. Maybe it'll be like ions or something. So like sodium ions. That's an example. So you have passive transport. It's passive. It'll just flow. So this means it'll go from high to low concentrations. We'll just write like high to low concentrations. So no energy needed. The cell can just do this naturally. Okay. We're going to highlight this. Wow. Okay, high to low. Okay, so what are some examples of this? So one thing that you can see is diffusion. You don't need any sort of protein, transmembrane protein for this. So what diffusion is if you have dissolved gases like O2 or H2, really, really small gases, they can just go straight through the lipid bilayer. Like you could have H2 flowing through here. It's so small it can get through these teeny tiny cracks, and it's nonpolar, so it's not going to like stick to stuff. It can travel through these like nonpolar tails, and that you can see that happen. So usually very small and usually nonpolar molecules. I know how to spell. Small and nonpolar molecules can pass through the lipid bilayer. Just to recap, diffusion, it's the very small, usually very small and nonpolar molecules. They can just pass straight through the lipid bilayer. So you don't need any sort of like help to do that. You can just do it. Cool. That is diffusion. But maybe we have facilitated diffusion. Pretty similar, but it just needs a little extra help. Facilitated diffusion. I can spell. Okay, so this one is pretty self-explanatory. It's diffusion, but it's facilitated. So we need a little extra help. So these are going to be for our like maybe larger or more polar molecules. Maybe they can't fit straight through the lipid bilayer on their own, but they can still get through the cell okay. They just need something like this like channel that'll like let them flow through. So usually a little bit larger molecules helped through with transmembrane proteins. So our transmembrane protein help move larger or polar molecules. And you can see a little bit of like, I say usually very small and nonpolar molecules diffuse. You can like occasionally see like slightly charged not slightly charged, slightly polar molecules diffuse, but like what you generally, as a general rule, it's going to be small and nonpolar for diffusion. But with the facilitated diffusion, we can have this guy allowing polar or larger molecules move through. An example of that is like water. So you have aquaporins, that's our fancy name for our transmembrane protein that helps with water, because water is polar, right? Help moving water. So aquaporin, that's a pretty good name because it's aqua, water, and pore, that's like a hole. So, your hole, that's an aquaporin. That makes sense. Love it when things are named in a way that isn't entirely confusing. So aquaporins, pretty self-explanatory, but transmembrane protein, right? And it's transporting water. But I don't have space. I'll go on to the next one. I'll just say transporting H2O. So that's what an aquaporin is. But this can also be our carrier, channel, receptor, whatever. An aquaporin is a type of channel. But as a general rule, facilitated diffusion, that's going to be done by carriers, receptors, channels. Okay, so this is from high to low concentration, right? That's pretty easy. Generally, that's increasing the entropy, which is favorable. But what if you want to have something, you already have it in low concentrations, but you want even less of it? You want to go against the concentration gradient, which is like that. The concentration gradient is like concentration, like you're going from low to high instead. So what if I want to go low to high? That sounds like it requires energy. And it does. So this is passive transport. Now we'll look at active transport. It does need energy. Because we're decreasing entropy, and that is not favorable. Okay. So we're going from low to high concentrations. So before, you don't need any energy. Now you do need energy. Okay, so you can see this in like carrier proteins. And there's 3 examples of this that we're going to talk about. So we'll see something called a uniporter. So we'll draw our little membrane again. We'll just have like a guy, just a guy. It'll look like this. So say we have like this guy, and it's going through our carrier protein. It's going to be traveling through the cell membrane, into the cell. Say this is outside the cell. We're going into the cell. We're going inside. And there's a lot of stuff in here already. So we're going against the gradient. That's difficult. That requires energy. So what a uniporter is doing, it's transmitting one molecule against the concentration gradient, one at a time. So we can see there's only one of them. There's hardly any around. We kind of want to go this way. So we have to use some energy. So the cell has lots of storage of energy, like around stuff like ATP. Do you remember what ATP is? We talked about it just a little bit before. It's called adenosine triphosphate, but you don't really need to understand that until cellular respiration. Okay, uniporter. But ATP is one of our nucleic acids, like DNA and RNA. But it has a lot of energy. So maybe we'll use some ATP, use some of that cellular energy to do something difficult like this, which is like take one of these guys and put them where there's already plenty of them. Like, why would you need to do that? But also, instead of a uniporter, you could have a symporter. Uniporter was one at a time. What do you think this one's going to be? Symporter 2 in the same direction. Bet you never could have guessed that. Okay, so there's lots of circles in here, right? Lots of circles. So we're going to have a circle, and we're going to add more circles. So that requires energy. But say we also have lots of squares. So many squares in this cell already. Pretend I'm also drawing them over here, but I'm too lazy. So like I'm just going to draw them over here. But like there's so many squares in this cell. Like we really don't need, well, we do apparently need more squares in the cell. That's why we're doing it. But it's like there's so many. They want to escape. So we got to use energy to put even more in there. You know, like if you're like herding some chickens, it's kind of like the chickens, if they're going to just naturally be about, they're probably going to be like evenly dispersed in the coop. They do kind of like to group up, but like just like generally, you know, around, if you got lots of chickens, they want to be evenly dispersed. But what you're trying to do is you're going to try to shove them all in one corner. That requires energy. Okay, so that's our symporter, similar to uniporter. These are both carrier proteins, but it's going to be transporting 2 in the same direction. Okay, so what is our third guy? So before we had 1 in one direction, now we have 2 in the same direction, because 1 in one direction, I mean, that's kind of how that works. Now we're going to have an antiporter, and this time it's going to be 2 in opposite directions. So what an antiporter does, we're going to have 2 going in the opposite direction. So we'll have a square going in here, you know, lots of squares, so many squares. Gonna require energy. But then maybe we have like triangles. They're abundant outside the cell. They want to go into the cell. They want inside. There's so many triangles. They want to be let in. But we don't want the triangles here. We hate triangles. So we're going to use energy to expel it from the cell. So that's what an antiporter does. It's like, we don't want you here. We're getting rid of you. But we do want this guy. We love square. This is not a very square-looking square, but we love square. Oh, that did not really help. Hopefully your square is better than mine. We love square. Come in here, square. So, just to review, uniporter, going to do 1 at a time. Symporter, 2 in the same direction. Antiporter, 2 in opposite directions. And so these are just carrier proteins using active transport. So you can see stuff like the sodium-potassium pump. I'm just going to abbreviate this as the pump, you know, sodium, potassium, which is an antiporter. It uses an antiporter. So this is something you'll see in animal cells. We use them like in our neurons. We need to have like this ion gradient where there's a ton of potassium in and a ton of sodium out. And like when you send an electrical impulse, it'll like change the gradient, and then that's how neurons work. I don't know. I'm not a psych major. But basically what that does is it uses 1 ATP, which is basically like the cellular dollar, you know? So 2 potassium in and 3 sodium out. So that's using an antiporter to put some in. So say this is a square. We use 2 potassium in, potassium ions, and then 3 sodium ions out. And that requires energy. Okay, but also, so you could be using, so this is just an example, the sodium-potassium pump. Oh, this is very good angle to be highlighting. Using an antiporter, we know what that is, to put potassium in and sodium out. But also, so you're right here we're using ATP, that's like our cellular dollar, our cellular currency. But we could also have coupled transport. And what coupled transport is remember how with passive transport, things will be going into the cell and that'll be increasing entropy, right? That's favorable. So it can be releasing energy, and what we can do is we can use the energy released by letting something into the concentration gradient to facilitate active transport and push something out that is against its concentration gradient. So that's just like the general idea. We use energy released from passive transport to power active transport. Okay, so that's a pretty cool thing that cells can do, kind of conserving energy that we're maybe using, because maybe we want, say like for example, there's a bunch of triangles out here, and it's like, okay, maybe we do want triangles in here, so we wouldn't be using an antiporter, but we let the triangles in, and that releases energy, and then we can use that energy to also push more squares in, because we love the squares. So many squares in here. So coupled transport, sodium potassium pump. You could look more into that if you are very interested in psychology and neurons and stuff, but personally, not necessary, probably. You might be asked sodium ions, potassium ions, what's going in and out, but chances are you just kind of have to know the general idea. What's an antiporter? How does that work? So you use energy released from passive transport to power active transport. That's pretty cool. Okay, so little review. We have a cell membrane. It's made mostly of phospholipids, and they're kind of moving and grooving around, kind of fluid, you know. If we want to increase fluidity, we can increase temperature, right? Temperature, or we can increase the amount of unsaturated fats. Not pictured. It would look, well, I don't really know how it would look. But you know, like these kind of straight chain tails, so like these straight lines are supposed to represent these nonpolar tails of the phospholipid. But if you had a unsaturated fat, it would be like there'd be a double bond, and then maybe if it was cis, it would like take up more space. So increased fluidity. Or you can have cholesterol, which is, at least in the strong, all big and taking up space, so maybe you won't be as packed. Okay. But also you could have phospholipids and polysaccharides bound together and making a glycolipid. Usually how these membrane elements, like a membrane protein, this membrane protein, how it works is this protein is going to be nonpolar, so it can stay in, like it can fit here, right? So this is a nonpolar region. I guess the way that we drew it, you can't really see, but like for a membrane protein, like maybe that's partially sticking into and out of the cell, or cell membrane. The part that's sticking in the membrane, that's going to be nonpolar, right? Because the membrane is nonpolar. That's like this whole bit, our lipid bilayer. The phosphate heads are charged, so that is like polarity. It's not really like, but it is. You know, like it likes polar molecules. But inside the where the hydrocarbon tails are, that's going to be nonpolar. And then the things that stick out of the membrane, you know, with our extracellular matrix, all the water and stuff, that part is going to be polar. But glycoproteins also have this like these polysaccharides. So we also see a polysaccharide here. So this is a protein plus a polysaccharide. Both of these are going to serve, the polysaccharides are going to be serving as ID tags for other cells to know, who are you? What are you for? Are you me? Or are you an invader? Or maybe, are you of the same cell type as me? Are we going to make a tissue together? And that's what glycolipids are so important for. So we have integral proteins that are in, peripheral proteins that are out. Well, technically this is inside the cell, but outside of the membrane, I mean. So remember, this is our membrane made of phospholipids. So outside, peripheral. But if you have something going all the way through, that's a transmembrane protein. And that'll transport molecules actively and passively. We talked about that. Kind of wish it was passively and actively in order, but it's okay. And we have carriers. That'll be like our uniporter, symporter, antiporter, but those can also be passive transport. These are all examples of active transport, but we can also have carriers that do passive transport. But the point is, it's one way with binding sites. So with our uniporter, it's going to like bind to the protein and then push it through. Channel proteins going both ways. Receptors, accepting signals or ligands and transmitting the signals to the cell. We'll talk more about that later. But it's less about, with the carriers and channels, it's more about like getting stuff that the cell needs into the cell or getting rid of stuff that the cell doesn't need or whatever. But with receptors, it's really all about the signal. Like it's less about the resources that the cell is using, and it's more like this is a message, and it's like it'll accept a message and be like, hey guys, did you know? Okay. So yeah, we'll have diffusion, facilitated diffusion. Aquaporins do facilitated diffusion, and remember this is like using a protein like this, a transmembrane protein. Diffusion, you can just go through the cell membrane with the lipid bilayer, you're chilling. So yeah, that's just like a basic idea of cell membranes and kind of our components. And with a little space here, I want to talk about how the vocabulary we use with the water of a cell and different types of cells. So different types of cells are going to want different like amounts of water. So we're going to be, I already spelled a very basic word wrong. Hopefully you know the word animal. Hopefully you know how to spell that one, animal. Okay, so we have animal cells and plant cells, and they're going to be a little bit different. It's almost like one is an animal and one is a plant. Okay, so we're going to see what does it look like when we're in a hypertonic condition. Oh, let's just write a little header. So this is going to be, we're talking about osmosis and tonicity, which are kind of like the same. Tonicity, there we go. Osmosis is like the flow of water, with our concentration gradient. Tonicity is like solute particles. So if you're in a hypertonic condition or situation, environment, whatever, say you put your cell in a bath, but it's not just any bath. We'll say this is the water. Does that look like water? Kind of. But this is like a salt water bath. Okay. So like this is salt water. So what do you mean it's salt water? So what's happening here in a hypertonic environment, there's going to be more solute outside the cell than in it. So the water is going to flow out. So in this, what we're drawing here, everything is going to be regarding water. And we're going to want to have 3 equal spaces. Mine aren't exactly equal, but to be drawing like our thing. So this is hypertonic. So hypertonic, water is going to be flowing out of the cell, and it's going to become shriveled. It'll shrivel up because, all the water is going to leave. Because remember, entropy, we're going to want to like kind of balance out our environment. So if there's more water in the animal cell, it's going to want to dissipate, be like even balanced out. And so in this salt water, there is more solute, and so all the water in the animal cell is going to want to even that out. So then we'll have a shriveled cell, and that's kind of sad. And so the animal cell does not like this. This is bad. It'll become crenated. Womp womp. Okay, what if we are in an isotonic environment? Well, in an isotonic environment, it doesn't have to be salt water, by the way. That's just like an example of what could cause this hypertonic environment. But in an isotonic environment, we're in like some sort of water-y thing or some sort of condition where there's no change or no net change. I'll say no net change. So like maybe you have water entering the cell, but it'll be the same as the water exiting the cell. And so animal cells, we like that. That's awesome. That face is creepy though. I don't like that. It's not that creepy. What if it? Oh, I don't know if that helped, but it's a happy guy. It's a happy animal cell. The water entering and exiting the cell is like the same, so we're chilling. It's like completely normal amounts of water. So we like that for an animal cell. But what if we're hypotonic? Hyper means like a lot. Hyperactive, a lot of activity. Hypertonic means tonicity is going to be really high in the solution that our cell is in. So with a hypertonic environment, there's going to be more solutes, and that's why the water rushes out, because it's like, oh, it's really like concentrated, and I want to make it less concentrated because it's less concentrated in the cell, so I'll rush out, and then we get a shriveled cell. But hypotonic is the opposite. hypoglycemia. I think it's like low blood sugar or something. But the idea is like there's not enough. There's less. Hypoactivity would be being like lethargic and tired. So within a hypotonic environment, the water there's going to be more solute in the cell than out. So water is going to rush into the cell. And then it's going to swell. So before it got shriveled up because it like lost all of its water. And now it's like ballooning because it's getting all of this water from outside from its environment. And that'll result in lysing, which basically means that it pops. Nobody wants that. So that is bad. Cell does not like that. The term for this is hemolyzed. So before, when we were talking about what happens in a hypertonic environment, it gets crenated. Now we're hemolyzed. These are both bad. We don't want to be shriveled. We don't want to be swollen. We want to be happy, isotonic. So that's great for an animal cell. We like to be isotonic. This is our little divider here. Okay. What about a plant cell? Well, what's the difference between an animal and a plant cell? Well, we don't know too much right now. We're going to learn later. But a key difference is plants have a cell wall. So we're in the same environment where it's like way more solute outside of the cell because it's hypertonic. So let's draw the cell wall like this. So the cell wall of this plant has kind of like, normally maybe it would be like a square. We can draw that for an isotonic environment. Like if it were isotonic, it would be a square. But now it's like shriveled up. And like the cell membrane is like really shriveled and it's like, ugh. But the cell wall is like kind of helping it. Like it's kind of keeping its structure. You know, it still has like this boxy form instead of being like this scraggly mess. But it does not like this. There's still water leaving the cell. It's still like the membrane shrivels. The plant does not like this. This is plasmolyzed. So that's the term for when it is hypertonic to a plant cell. It gets plasmolyzed, not happy. Okay, well, what about when there's like an equal exchange of water into and out of the cell? Because we're isotonic, so it's about the same. So the cell wall, cell membrane, everything's going to be like pretty even. Everything is making sense. So just to clarify, so this is our cell wall. And this is our membrane. The cell wall of a plant cell is like on the outside. That's how all cell walls are. It like guards the membrane almost. So the membrane, we're chilling. But plant cells don't actually like this. This is actually kind of sad for them. This is a flaccid cell. Because what plant cells really like is they like having lots of water. So let's say we have so much H2O, it's getting swollen, you know? We have lots of water being pushed or going into the cell. So we have our membrane, it's like pushing up against the cell wall. We're all like a big fat balloon. And what plant cells have is a central vacuole full of water. And so when all this water is rushing into the cell from this hypotonic environment, this vacuole is going to like push outward, like the vacuole itself, not the water. It's going to push out, and it's going to create this thing called turgor pressure. So we'll just say turgor pressure, and creating a turgid cell. And plant cells really like that. That's good for a plant cell. So they're not happy when they're shriveled or when they're flaccid. They're happy when they're turgid and they have lots of water. And what keeps them from popping or lysing like an animal cell is this really strong cell wall that's holding everything together. And this is the optimal environment for a plant cell. Obviously, with an animal cell, there's no strong cell wall, remember, made of cellulose. Keeping everything together so it'll pop, and that's not good. Nobody wants to be popped. So isotonic, great for an animal cell, not so great for a plant cell. It wants to be hypotonic. Okay, so that's just a little bit of a review. Not a huge part of cell membranes, but they do play a role here, and it is really relevant to the entropy that we talked about before. So yeah, that's our discussion of cell membranes. Very important to the general keeping a cell together as a single cell. And you're like, well, what's in a cell? What distinguishes one cell from like all the other stuff, you know? Like, why can't you just have one big, like, instead of individual cells, why can't you just have like all of your stuff out and about in your organism? And we're going to talk about that. We're going to talk about what's in a cell. We're going to talk about what they do. What's the point? But not yet. Next lecture. Tune back in for Into the Cell. Goodbye.